All right, let's dive straight into this explainer. We're going to equip you with exactly what you need to master, honestly, one of the absolute most essential techniques you'll ever perform at the lab bench. Whether you're a student prepping for a practical lab or just someone fascinated by how we actually decode the building blocks of life, you are absolutely in the right place. So picture this. You've just knocked out a PCR reaction, making thousands of copies of a target DNA region. Or maybe you're doing some DNA cloning and trying to paste a specific gene into a circular plasmid. You obviously need a practical way to check your work, right? But how do we actually visualize these microscopic, invisible fragments to verify our results? Well, we're going to treat this explainer like your own personal step-by-step lab masterclass. Here is the agenda. 1. Seeing the invisible DNA. 2. Gathering your lab materials. 3. Setting up the gel. 4. Running the electric race. And 5. Visualizing the results. Section 1. Seeing the invisible DNA, the foundation. Gel electrophoresis. It's a crucial lab technique used to separate macromolecules, things like DNA, RNA, or proteins. The really cool part here is that this technique relies entirely on physical properties, specifically size and electrical charge, to sort molecules that we otherwise couldn't see at all. We're essentially forcing these microscopic fragments to reveal themselves by making them run a very specific kind of race. Section 2. Gathering your lab materials, the ingredients. Being organized is everything at the bench, so think of this as your lab recipe checklist. To make this work, you need your agarose gel, a buffer solution, usually TAE or TBE, which is basically just salt water, a power supply, some sample loading dyes so you can track your progress, a DNA ladder to act as your reference marker, and a staining agent. Now, the real star of the show here is the gel itself. As the name hints, you'll be working with this slab of jello-like material. It's usually made from dry agarose flakes, which are polysaccharides. You heat those flakes up in your saltwater buffer, let it cool, and it forms this solid, slightly squishy matrix held together by hydrogen bonds. Basically, you've just created a microscopic, highly porous obstacle course for your DNA. Section 3. Setting up the gel, the protocol. So here are the 5 exact steps you're going to execute in the lab. Number 1, prepare and cast the gel. 2, load your samples into the wells. 3, apply an electric current. 4, let those molecules migrate. And 5, stain and visualize. It's a super reliable sequence that takes you from a completely invisible sample to a glowing, measurable result. When you're setting up the apparatus, precision is key. You place the gel right into the main body of the gel box. The trick here is to make sure the salt-containing buffer just barely covers the top of the gel so it can conduct the current properly. And this is crucial. You absolutely must position the end of the gel that has the pocket-like indentations called wells towards the negative electrode. Once that gel is snug in the box, you'll carefully transfer each of your target samples into those tiny wells. And hey, don't forget to reserve one well specifically for your DNA ladder. That ladder is your standard reference. It contains DNA fragments of known lengths, so you definitely want to pick a commercial ladder with good size coverage for whatever it is you're expecting to find. Section 4. Running the electric race, the mechanics. Your samples are loaded. You flip the power supply on, the current starts flowing, and the race begins. But why does turning on the power actually make the DNA move? Well, it's because DNA molecules inherently carry a negative charge, thanks to the phosphate groups in their sugar-phosphate backbone. Because all the DNA has this negative charge, applying an electric field forces it to run away from the negative pole and race straight through that squishy matrix toward the positive electrode. And this is the brilliant part. Since all the DNA molecules share the exact same charge-to-mass ratio, the gel acts as a sieve to separate them purely by size. The large DNA fragments get all tangled up, so they move slower through the pores and stay near the wells at the top. Meanwhile, the small, nimble fragments easily outpace the large ones, traveling way farther down the gel. But listen, here's a quick, real-world warning straight from the lab bench. You've got to pay close attention to your timing. As our source points out, if you leave that electric current running for too long, your shortest, fastest pieces of DNA will literally run right off the end of the gel and get totally lost in the buffer. Nobody wants that. Section 5. Visualizing the results, the reveal. Assuming you timed it perfectly, the race is over and the power is off. But you still can't see the DNA with the naked eye. To actually reveal your results, you have to stain the gel with a DNA-binding dye, like ethidium bromide or maybe a safer modern alternative. Once it's stained, you slide that gel under a UV or blue light and suddenly, boom, those hidden DNA fragments brightly glow. Looking at the glowing gel, you're going to see what we call bands. Now, keep in mind, a single DNA fragment is way too small to see by itself. You aren't looking at just one piece of DNA. What you're seeing as that glowing, well-defined line is actually a massive group of identically sized DNA clones that have all traveled together to that exact same spot. Reading the gel layout is surprisingly straightforward. Just remember this rule. Bands closer to the wells, up near the top, are your larger fragments. Bands positioned farther down, closer to the positive electrode, are your smaller fragments. To turn those glowing lines into hard data, you look back at that DNA ladder you loaded earlier. By lighting up your sample bands horizontally against the known yardstick of the DNA ladder, you can pinpoint the exact base pair length of your product. So for example, if your band lines up just a bit below the 1,000 base pair mark on the ladder, you can confidently identify it as roughly a 700 base pair fragment. Ultimately, why does all this matter? Well, this simple, squishy matrix is an absolutely foundational tool in biology. You're not just learning a basic student lab chore here. You're learning the exact techniques scientists use to verify PCR products, conduct genetic testing, and even solve crimes with DNA fingerprinting and forensics. It's a wildly powerful tool. So, you now have the practical steps to visualize the fundamental building blocks of biology, and you understand exactly how size and electrical charge can sort invisible molecules. The real work begins now. What mysteries are you going to uncover at your lab bench next?